Okay, hi everyone. My name is Khong, and I'll be moderating this session, which is for complex traits and variant resolution. So we have an exciting panel of 3 speakers today. And I think before we proceed, I just have a few housekeeping messages. So the first is our sponsors, PacBio, as well as LifeBit, warmly invite you to their evening networking events immediately following day 2, which is tomorrow's program. So not today. Don't go to the restaurant. You won't see anyone there. But tomorrow, after tomorrow's program, the PacBio and LifeBit will have separate networking events. So please join them for a special evening to connect with industry leaders, experts, and professionals. For the PacBio event, please visit their booth to register your interest. They still have places, but I understand that they're running out. And for the LifeBit event, please adjourn to the Escape restaurant by the poolside. So that's over here. Yep. So that's for tomorrow. The other thing as well, for this session, we'll be using a real-time web-based application known as Pigeonhole Live, where you can post and vote for questions using your smartphone, tablet, or laptop. Please participate in the Q&A session by logging in to pigeonhole.at. www.pigeonhole.at, and key in the password PIC in all caps 24, PIC 24. You may send in your questions or vote for existing questions, and this information can also be found on the LED screens. All right. So without further ado, I would like to introduce the panel of speakers that we have for today. The first speaker that we have is Prof Wong Limsoon from the National University of Singapore. Prof Wong is a professor in the School of Computing at the National University of Singapore. He's also an honorary professor of pathology in the Yong Lu Lin School of Medicine at NUS. Limsoon is a fellow of the ACM, and some of his other international research awards include the 2003 FER Asian Innovation Gold Award for his work on treatment optimization of childhood leukemias, and the ICDT 2014 Test of Time Award for his work on naturally embedded query languages. In recent years, he has been pioneering protein complex-based approaches to overcome, in simple yet refined ways, the consistency and coverage challenges that have plagued untargeted proteomic profiling. And today he'll be speaking to us on single-cell RNA-seq dataset integration without loss of unique rare cell populations. So, Prof. Wong, please.
Good afternoon. Although when we think about complex traits, we tend to think about genetic mutations, but today I go a slightly different route because many diseases are not mutations that you are born with, but they develop over time, the somatic ones. And to figure out what are these, you don't want to wait too late until your whole body is full of these mutated somatic mutations. You want to catch them as early as possible. This is a very dynamic process, and how to catch them or discover these mutated cells as early as possible is what I'm addressing today. To give you an example, or the technology that is being used today is probably single-cell sequencing technologies, and it has one very basic and fundamental problems to almost all high-resolution genomics. Here I have the 2 pictures displaying single cells from 7 samples. Actually, these 2 pictures are exactly the same data, I just color them differently. Let me first explain this picture for some of you who are not familiar with single-cell sequencing. So every dot in this picture is 1 cell, and the position, the XY coordinates, represent the expression profile of these cells, okay? So look at the left, the cells are colored based on the types of cells that you will see. For example, the green cells, the greenish, brownish yellow, is the CD4 cells. And you will see in the center, there are these 5 green fingers. They are all CD4 cells, the same kind of CD4 cells, but they are in 5 different places in this picture. So to you will think like maybe there are 5 different kinds, in fact, 7 different kinds of CD4 cells, but actually there's only one. So what is happening here? Now let's look at the picture on the right side. You see the same 5 fingers, they're colored differently. This time, I color the cells based on sample of patients. Each patient has its own color. And you will see that the 5 green fingers correspond to 5 different patients. The same cell is the same, but it's the same 5 different patients. In fact, even if I have the same patient, I run the single cell sequencing 5 times on this guy, I will get that 5 fingers. Okay? So these are not biology. These differences in their profile, in the position, has nothing to do with biology, but very much to do with the technical effect we call the batch effects. Whether I sequence this person on Monday or Tuesday, the same cell have a different position. So how would you even do correct analysis in the presence of this kind of technical effects? Yeah? So this is the problem I want to deal with. And you want to analyze this data, the first thing you have to do is to remove that technical effects. And there are many tools that you can use today to remove this. And I show, actually at the top, I named Seurat. This is probably the most popular because it has a software that's very easy to use. You don't need a lot of brain to use it. And, and it does look like very successful. So for example, all my green cells has now grouped into one place, right? So it makes sense. I've successfully dealt with the batch effects, apparently. Is it real? Actually, it's largely true, except for this issue. Over here, now I have one sample, and the different type of cells is still plotted based on their profiles. Positions still represent profiles, dots still represent individual cells. And I have highlighted some blue ones and red one and green ones on the slides. Obviously, let's take the blue one. It is isolated from the rest, right? So it is a different types of cells because this is one patient. I have no batch effect, one patient. If I integrate this using Seurat or any other tools with, in this case, 16 other patients, and then you ask, where is my blue cells? They are there in the original, but after I integrate, there is no more blue cell. They disappear. What is happening? What is happening is because in that integration, tools like Seurat, when it is removing batch effect, it has also removed some biological effect. It removed the differences between these blue cells and merged them with some other cells. So you lost that. Now, is this loss important or not? That depends on what you're looking for. If you're looking for cells that let me call them pre-malignant mutated cells, cells that mutate before you get cancer. You know, you don't get cancer overnight, right? It develop over many days, many months, many years. So initially, you get some cells that are mutated, and then these cells accumulate other mutations until it accumulates all the important hallmarks of cancer, then it blows up, right, and multiply like mad. Yes, but before that, no. That cell will remain in a very small population because, after all, mutation tend to make that cell a bit less efficient than the original because it's easier to spoil things and make things better. Yes? So they will always, these early mutated cells will always be a very small subpopulation, and they will look quite similar to original parental cell, the unmutated ones. And 90% will be the unmutated one, and then this 1, 2, 3% will be these mutated versions. And those, because they are so similar to their parental cells, when the batch effect removal is done, they tend to remove that small differences caused by these genuine somatic mutations. Okay, those you will lose. So if you're looking for cells that represent very early pre-malignant mutation, you will find nothing using single-cell technology unless you're very lucky. Okay, so this is the problem of when you remove batch effect, you remove along with it some important biological effects that you're looking for. Okay, and that's the problem I want to highlight here. First message to take home, be very careful with your single-cell analysis and with the batch effect removal. Second, of course, I want to show you a possible solution to this. This is map batch developed by my postdoc. So it makes use of some modern AI development called autoencoder, but we use autoencoder in a very special way. For those who are not familiar with this technology, an autoencoder has two components. One we call an encoder, the other we call a decoder, and these two components are connected by a bottleneck. So that's very interesting, because if you have input X going into the encoder will spit out an encoded version, what we call the latent space, via the bottleneck. Because the bottleneck is small, it has to throw away things, okay? And then the decoder will take this reduce latent space and decode it to regenerate the original input. That's what the decoder does. And so since encoder is squeezing information through the bottleneck and has to remove information, the decoder has to reconstruct that information that you have removed. So what this autoencoder is doing is basically deciding what information to remove so that it is easier for the decoder to reconstruct. Yes? So you have to remove things that are easy to reconstruct later. Most people that I've seen using this one to deal with batch effect, they do it in the wrong way. When they train, this is AI, you have to train them. They train them using all the data batches to train the autoencoder. So you have seen my 5 fingers. They're all CD4 cell, the biology is the same, but yet they're in different position. That means, and this position are batch effects. So that means the batch effect dominates the biology if you look at all the batches of data at the same time. So what would this autoencoder throw away when they squeeze the information through the bottleneck? It has to throw away things that is easy for it to reconstruct. The biology represents a smaller change than the batch effect. So what it is throwing away? It's throwing away the biology and retaining the batch effect. So you don't get a very good solution using this. Okay? So given what I just told you, how should you train this? Well, I guess one way to train it will be to train an autoencoder using only one batch. Because within that batch, all the cells have the same batch effect or similar batch effect. So the batch effect becomes the least important information, right? So it will be the fewer information that you can throw away, and the decoder can easily reconstruct. Okay, so trained it using only one batch. And that is very important. Now I have, let's say I have just 7 patients, I have 7 batches. So what do I have? I train autoencoder only one batch. What happened to the other 6 batches? That requires a very good computer science thinking. So we will train 7 autoencoder, 1 for each batch. Each autoencoder is specific to that batch. So each autoencoder will learn to throw away or learn to retain the biology of its specific batch, but throw away all the other information that is not compatible with the biology of that batch. Okay, once I've done that, then every data that I have, every single patient, I'll give it to all 7 different encoders. So what will happen? I give it to autoencoder 1, all the batch effect in, let's say this data is from batch 2. So all the batch effects from batch 2 will be thrown away because it's not compatible with the biology of batch 1. And also, all the biology, all the unique cells in batch 2 that is not present in batch 1 will also be thrown away because they're not compatible. And after that, the decoder of that first autoencoder will inject the batch effects of batch 1. So what I get is what this patient from batch 2 would have looked like if he was sequenced in batch 1. Okay, but now that same patient, I'm also giving it to the autoencoder number 2. Yes, so it's the same batch. So it will come out, will be reconstructed perfectly, including those cells that are unique to batch 2. And similarly to all the other batches. Now you may say, okay, now one patient suddenly becomes 7, right? What am I going to do? Now what is very interesting thing that we took advantage of is that the next step that you want to do is to cluster, right? You want to put patient that looks, or cell that look the same into the same cluster, and cell that look very different into different cluster. So to do this, you need to compute the distance between every pair of cells. Yes, how do you compute distance? You take my cell X, because I have 7 autoencoder, you have 7 output, 7 components. Cell Y, a different cell, also have 7 components. But these components are very special because the first component is what this cell would look like if it was done in batch 1. Yes, the batch effect of batch 1. Cell Y has, first component has batch effect of batch 1. When I compute the distance, I take component Y. So I take component 1 of X minus component 1 of Y, right? Take the square plus component 2 of X minus component 2 of Y, and so on, right? And then take the square root. That's geometric distance, right? So what will happen when I do that minus? The batch effect cancel each other. So my distance, even though all my data has batch effect, the batch effect cancel each other when I determine the distance. My distance is a pure biology difference, distance. Okay, the batch effect disappear from my distance calculation. And the other interesting thing is, so I can resolve batch effect. And the other interesting, how do I retain the unique cells? Right, let's say to batch 2. That happened because I have component 2. The distance in component 2 has the biology of batch 2. So I don't lose these unique cells that are unique to batch 2. So I solved the 2 problem. And if you look at the output, here, this is on the upper left corner is output of the integration of that 7 patient produced by this technique, map batch. You see, for example, my blue, red, green for that particular patient are retained. They don't disappear. Whereas you look at Surat or some other popular technology, they disappear. Okay, so this is how you resolve the batch effects, but retain the biology that's unique. Now let me show you what you can do with this example. I'm not sure how much time I have. 5. Oh, okay, I'll do it very quickly. So this is an example of multiple myeloma. I think it's reduced data. And when he initially did the single-cell sequencing, he saw nothing unique to what he wants to look for, which is cells that are associated with progression of that leukemia. And when we processed using MapBatch, actually we found very distinct clusters, including those clusters that you do not know what they are. You cannot annotate them. And this is cluster 26. And cluster 26, this type of cells are very interesting. Because if I look at his patients, those that have contain a lot of these unknown cells, and those that have nothing, this is the survival difference. Right? And then once we have that, we know this cell is important. Then we look at the expression profile of this group of cells and identify what is special, what genes are special. So we determine the marker, and then when he applied this marker to subsequent 2 cohorts, the same split was found. Okay, so that is one interesting thing you can do with this unique cell. So you know which are the cells that are mutated very early, and you know what are the signature. Right, another example, this is colon cancer from David Worshop. He read a paper from Nature Medicine, and that paper basically discarded, I believe, like more than half of the single-cell output because they didn't know what it was, or some was discarded, they don't know what it was, and some were discarded because some quality threshold, arbitrary one, were not met, right? So we run this, set the data up. In particular, he was not happy because after discarding all those things, what remains, he couldn't see any stroma cells, right? So it cannot be. So we look at the data for him, and we found very large number of stroma cells, and they all cluster very together, not scatter. So, and then for this stroma, we subcluster them. These are the clusters. And then over there are two clusters that are very interesting. One is cluster 14, the other is 17. Why are they interesting? Because the colon is very long. So you can basically chop one part of the sample, one part of the colon that is normal, one part that are polyps, one part that is the tumor itself, right? And then we look for cells, cell types that are present a lot in the normal and then decrease in polyps and decrease even more in the tumor, and the other way around. And these two clusters that we found exactly have that behavior. Of course, I do not know what, I'm not a biologist, I do not know what the cells are, and we hope David's group will be able to figure out what they are. Okay, so I think my time is up, so I will stop here. Okay.
Thank you very much, Prof. All right, so next we'll be hearing from Dr. Jimmy Lee, who is from the Institute of Mental Health. Dr. Lee graduated with his medical degree from the National University of Singapore in the year 2000 and attained specialty accreditation in psychiatry in 2009. Under a scholarship from the National Research Foundation and Ministry of Health in Singapore, he completed the Master of Clinical Investigation in 2010. Dr. Lee furthered his clinical and research training at the University of Toronto and Centre of Addiction and Mental Health, Toronto. Presently, he is a practicing psychiatrist at the Institute of Mental Health and an associate professor at Lee Kong Chian School of Medicine, Nanyang Technological University. Dr. Lee's research motivation centers on the improvement of outcomes and recovery in people with serious mental illnesses. He adopts multimodal phenomics approaches to bridge the gap between clinical science and neurosciences with a vision to improve clinical decision-making and treatment modalities. Additionally, he is interested in the development and application of digital technologies in mental health care. Dr. Lee has received several research grants and awards, including the NHG Clinician Leadership to Research Award in 2008, NMRC Transition Award in 2012, NHG Clinician Scientist Career Scheme in 2015, NMRC Clinician Scientist Award in 2018 and 2023, and the NHG LKC Clinician Scientist Award in 2021. Today, he'll be speaking to us about realizing the dream of precision medicine in schizophrenia. So without further ado, Dr. Jimmy.
Thanks, Weng Khong. Really much appreciate you reading out the entire thing. I didn't expect that. So good afternoon. Thank you, everybody, for coming to Little Dream of Mine. I'm not the only talk in this 3-day conference that used the word dream. There's another talk in the next 2 days that use the word dream. That tells you that it hasn't happened yet. All right, so I will not introduce myself further. Okay, great. So some conflict of interest declaration. So the condition here I'm talking about is schizophrenia. So we talk about complex traits, and I think probably, arguably, and I'll put my hand out there, my head on the chopping block, there's probably no complex disorder or disorder as complex as neuropsychiatric conditions. So this is a condition for which I have been working alongside persons who have been afflicted with it for almost the last 20 years of my career. And actually, this data set I'm sharing today has been gathered over the last 5 to 6 years only. So most of us will know schizophrenia, but we probably do not know the family of conditions called psychosis, where an individual loses touch with reality. So schizophrenia is arguably the most well-known condition that is listed under the umbrella of psychosis. It doesn't spare low-income, high-income Asia-Pacific countries. The worldwide prevalence is roughly really about the same. It's about 0.7%. It's also one of the leading causes of disability worldwide. It's characterized by a series of behavioral syndromes and things that we can observe, from perceptual disturbances, disorganization or thinking disturbances or beliefs, even cognitive impairment, as well as some deficits in terms of their emotion expression and their motivational levels. It's not solely a brain or mind disorder. Patients with schizophrenia do have at least a shortened or reduced lifespan of 15 to 20 years. The odds of death from cardiovascular disease is actually about at least 2 times that of a normal population. All right? And if you are in my generation, you probably have watched this show called A Beautiful Mind, played by Russell Crowe when he was much younger. Really much younger. Okay, so in all treatment guidelines for schizophrenia, antipsychotics form the mainstay for treatment. And this little PubMed figure is just a cheeky way for me to let you know since 1966, but actually the first antipsychotic was discovered in the late 1940s, early 1950s. That's where it became FDA approved, till today in 2024. Now, unlike most of the other conditions or even pharmacological agents that sees exponential growth, you see a slightly downward trajectory for antipsychotic development, as this is not due to COVID. In my hospital's formulary today, we actually have 16 antipsychotic agents that we can use, broadly classified first, second, and third generation. Those in underlying are medications that you can give orally or intramuscularly. If you do a search in Wikipedia, I last counted 69 antipsychotics that were ever approved by any regulatory agents for use. Now, to the uninitiated, that would be like, oh, wow, that's a huge arsenal you can use. For sure, you must have solved the problem of schizophrenia. But basically, more doesn't mean good. More actually simply indicates to us that there's a lot that we do not know. And the chart on the right is, I believe in 1975, this is probably hand-drawn, and it's still valid today, that spawned the hypothesis or the dopamine hypothesis. And basically, it states that the agents then in the era in the early 1970s appear to be very dopamine-related in this relationship with schizophrenia treatment response. And the cartoons on the left tells you the 4 dopamine pathways that we talked about. Of the 4 pathways, the one that we are hoping to kind of target to remediate or to ameliorate the symptoms of schizophrenia is the mesolimbic one. The other 3 pathways basically connotate side effects from medications. Why? Because our antipsychotic agents are not smart bombs. They go and they bomb the whole brain. Every single thing that has got a dopamine receptor, I block. I don't care whether you are from the limbic system, nigrostriatal, or whether you are in the prefrontal cortex. You look like a dopamine receptor, I'll block you. And that explains a lot of the side effects that we see with the present agents today. Now, then of the 69 agents that I talked to you about and the 16 that's available in IMH, when we start taking a look at the efficacy outcomes, and it's quite surprising actually to a lot of practicing clinicians, there's actually no difference in efficacy. None of the new generation antipsychotics or the old first generation antipsychotics show clinically significant difference in efficacy profile. But what they do show quite significantly are differences in side effect profile. So the older generation agents, which tend to be having more binding affinity with postsynaptic D2 receptors, they tend to cause a lot of motoric side effects, extrapyramidal side effects, tardive dyskinesia, which is a cartoon, or rather, which is the picture that I'm showing here of this lady who suffers a very disabling side effect called tardive dyskinesia. The second generation agents or the newer agents, because of either their histaminergic kind of receptor profile, tended to cause a lot of cardiometabolic side effects, obesity, diabetes, even things like hyperlipidemia. But really, when it comes to choices of antipsychotic other than our preconceived notions of what my senior taught me in terms of which is the best agent to use, really when it comes to choice of medication, isn't really rooted by a lot of science, whether I chose drug A, drug B, or drug C. And by and large in Singapore, because of the healthcare system being co-pay, cost tends to factor in as a significant component. Why is that so? Because this is not a flu I'm treating for 5 days. This is a lifelong condition. Whatever I prescribe, this person is going to be on it for the rest of his life. Now, moving along, now we talk about antipsychotics. Earlier, previous slide, I showed you how there's actually no differential difference in terms of the efficacy, but there's a particular group of individuals, estimated about 30%, who don't respond at all to any of these antipsychotic agents. This is what we call treatment resistance. And what is so interesting about this group, or what is so impactful or important about this group of individuals, is that they are responsible for more than 10-fold increase in direct healthcare costs. I'm not even mentioning the indirect healthcare costs. How many of them get institutionalized, requires permanent care settings as a result of their persistent kind of symptoms. In this particular group of individuals, the only agents out of the 69 in Wikipedia that's indicated for use is this thing called clozapine. Unfortunately, for various reasons, either because of their side effects or the intense monitoring profile, it tends to be significantly delayed. And in the study that we published, or rather from our own data set, what we found was the average years of delay was as long as 10 years. And after almost 6 antipsychotic trials. In our treatment guidelines across the world, typically clozapine is recommended as a third-line agent. If you fail 2 trials of antipsychotic, you qualify for clozapine. But somehow in clinical practice, that doesn't really seem to happen. So we started to ask ourselves, why is that so? And we say something about the doctor's training, they're not informed, or is it something about the particular agent? So we collaborated with Hong Kong, and then we actually asked the clinicians how much they know about treatment resistance and what do they actually do when it comes to treatment resistance. what we found was that, fortunately, the clinicians are familiar with the guidelines. They know when to use, theoretically, clozapine. But most of them, when they face with a patient with treatment resistance, would choose other off-guidelines kind of treatments. For example, combined antipsychotics. I give 2, 3, 4 antipsychotics. I add on mood stabilizers and other agents to try to ameliorate the symptoms. And when we ask them specifically about clozapine, why don't you use clozapine? Now, they all know that clozapine is effective. They are also very cognizant that clozapine is underutilized. But what they cite then is the barriers in terms of monitoring. They felt that the patients actually didn't want, and there's an administrative burden as a result of lifelong neutrophil monitoring. So interestingly, in the naturalistic study, and this is the one done by Professor Akil from Toronto, and this data has been also replicated in other data sets. Earlier, I talked about clozapine being a third-line agent, and in this naturalistic kind of clinical cohort, the first trial, antipsychotic, second trial, antipsychotic, third trial there will actually be clozapine. Now, the response to the first trial is 75%, the next response drops to 16%, and when the person becomes resistant and takes on the third trial, which is clozapine, the response goes up to 75% again. This is a very unique profile that, like I said again, not unique to Canadians. We see that in Europeans as well as in the Asians as well, but not often talked about or studied. By using this and other data between myself and my mentor, Professor Gary Remington, we kind of postulated pharmacological subtypes of schizophrenia. Without needing any scans or imaging, basically what we do is through our naturalistic pharmacological guidelines, we have managed to pigeonhole or categorize patients based on their antipsychotic treatment profile. If we believe the previous Lancet study, all antipsychotics are the same, clozapine is the only one that's unique, then a very simple kind of decision tree, you're either antipsychotic responsive or resistant, and among those who are resistant, if you're exposed to clozapine, you either are responsive or you are resistant, all right? And these form the 3 pharmacological subtypes that we further extend our study. So this is a little short publication that we did late last year. In the data set that we have, we tried to take a look at the architecture of treatment-resistant schizophrenia, adopting whether polygenic risk scores can help us to identify them. And interestingly, because, this is Singapore, our samples are uniquely Asian, we wanted to take a look at how the European kind of data set may be relevant or useful. So in this particular study, we had about 142 well-characterized patients who were exposed to clozapine. So this is like definitively treatment-resistant. We have about 715 patients who never had clozapine before, but they probably had multiple antipsychotic combinations or they were responsive to antipsychotic agents. And we wanted to compare the two groups. So this is more or less the bioinformatic pipeline. We genotyped them on the Illumina microarray, imputed against 1,000 genomes, and we computed the polygenic risk score either using the East Asian ancestry or actually the European ancestry that was published. And we used p-value threshold for the PRS calculation. So if I can direct your attention to the first column on schizophrenia. So the first thing we did was to compare all schizophrenia versus healthy individuals with no schizophrenia. Looking at if we use the East Asian ancestral background or the European data set to compute the polygenic risk score. So you can see that both kind of ancestral backgrounds, no significant difference. I think we can generally identify schizophrenia quite well. If you look at breaking down the schizophrenia into the non-resistant versus the resistant subset, so what you see is that in the non-resistant subset, again, quite comparable between the European or the East Asian polygenic risk scores. But we zoom in on those who are treatment resistant, and that's where the ancestral background starts to show a slight difference, favoring East Asian background in applying in an East Asian data set that has got treatment-resistant schizophrenia. And when we looked at within disease, within schizophrenia, what ancestral background can have an impact of in terms of identifying treatment resistance. So the panel on the left there shows when you use the East Asian data set, we have an odds ratio of 1.5. Not a trivial or small effect size measure. But when you use the European data set, basically none. There's no significant findings that we can identify. So this is a kind of an interesting finding that we have, and one of the first Asian studies to show the importance of using the appropriate ancestry background. But this is, again, not a new problem. We are just trying to show the impact of this challenge. And most of us will be quite familiar with this diversity gap in genetics. And again, on the panel on the left, you will see the UK Biobank showing exactly the same kind of problem across the different ancestries. East Asian, which is the kind of data set that we use, showing how we actually differ significantly if we compare to Europeans. So this is the slide on complexity. So schizophrenia is a complex phenotype. Put it simply, this is a behavioral syndrome. So we are actually classifying or identifying based on certain sets of behaviors. The behaviors can go on based on psychopathology, which is a 30-item instrument you see on the panel on the right. They can categorize them further into positive symptoms, negative symptoms, conceptual disorganization, or even mood. And on the left, on the bottom left, you will see different cognitive trajectories. Earlier, I mentioned how cognitive impairments is a significant component of schizophrenia. And here you see various cognitive trajectories, including those who are cognitively preserved, as well as those who seem to have a very poor cognitive profile from the very beginning. So how do we then work around, a complex phenotype such as schizophrenia? Now, when it comes to treatment, if embracing the previous slide in terms of the complexity of the phenotype, then how can we as clinicians anticipate a single molecular compound to target such a complex phenotype? And I think this I suppose, fallacy is something, or this nightmare is something that we need to wake up from. And the concept of antipsychotic as a magic bullet, that we can use a single antipsychotic that targets primarily D2 receptor antagonists and yet try to target positive, negative cognitive impairments as well as things like negative symptoms. In the interest of time, I'll move on. So if I were to use the same concept that we have in psychiatry, or rather using antipsychotics, and transfer that over to diabetes, a field that most of us are quite familiar with, I'm sure you don't hear endocrinologists using the word anti-diabetic agents. So they go very specifically by mechanism of actions. And why do they want to do that? Because there are certain benefits. Now, what you can do now is rational polypharmacy, take it as you will. You combine medications based on certain mechanism of actions to try to augment or enhance the symptom profile. The clinical outcomes. Moving on to the last part of my talk, here I'm going to talk about clinician perception. Now, while we talk about use of polygenic risk scores, any form of genetic risk scores in clinical practice, at the end of the day, the patients are not going to be one ordering their own polygenic risk scores. You have to be the clinicians. So in the earlier survey, we asked the clinicians about their attitudes towards treatment resistance, their attitude towards clozapine, and you can see some considerations or concerns clinicians manifest or report. So here, when we ask clinicians, if I do have something that is a pharmacogenetic risk scores or pharmacogenetics for psychiatry, would you use it? Now, most psychiatrists know, maybe this is social desirability bias, that psychiatrists should order pharmacogenetic risk scores. They can see the value of it, but what they have cited quite clearly in the survey is they don't really know how to interpret or how to use it. They want something that's more definitive in terms of guidelines. They need to know how it impacts directly their clinical practice. So these are areas, regardless of whichever genetic risk scores we're looking at for whatever condition, something for clinicians to consider and bear in mind when we want to translate or implement genetic risk scores in clinical practice. So this is my last slide, and I really like this picture for Singaporeans who do national service. You're very familiar, right? Some of us remember having empty casings thrown at our helmet. So this is called, when we go range, we do zeroing, right? You need to be accurate and all. So when we talk about precision medicine, a lot of us are talking about how we can be precise and keeping all our identified areas or targets or biomarkers together that's close to the phenotype of interest. Now, in this particular study that I showed you in terms of treatment resistance, the genetic markers may or may not have anything to do with the biology of the disease. But what it does is that it appears to have a very strong association in terms of predicting the outcomes of that particular disease in relation to treatment. So maybe, lucky as you're not, it may be in the top right corner where it may have low accuracy in terms of a disease itself, in terms of schizophrenia prediction, but very high precision. And what we need to know is see how we can actually kind of zero to bring this back in terms of the clinical outcomes that we want to target. So in conclusion, what I want to hope to transfer the message across is that the genetic burden of schizophrenia, here I'm talking about overall schizophrenia, may in part actually explain resistance in schizophrenia. Polygenic risk scores may help in early identification and detection of treatment resistance, hopefully facilitating early access to clozapine, a point that Patrick talked about in the earlier session, where he used polygenic risk scores for breast cancer to nudge patients to do screening. Here I'm using polygenic risk scores for treatment resistance or response to nudge clinicians towards early access to clozapine. And here I also want to impress about the importance of using the appropriate ancestral background when you want to do things like genetic risk scores. We need to have increased precision in phenotype. While we talked about schizophrenia broadly, there are also sub-syndromes within it for us to consider. And not to forget the end users, and in this case, in terms of deployment, implementation, you're looking at actually the clinicians and users that are important to factor in. Thank you.
Right. Thanks, Dr. Jimmy Lee, for your talk. So our third speaker for today is Dr. Ammira Al-Shabeeb Akil from Sidra Medicine. Dr. Akil leads the Sidra Medicine's Genetic and Metabolic Clinical Research Program and specializes in genetic and metabolic diseases. She oversees the Precision Medicine of Diabetes Prevention Lab at Sidra Medicine in Doha, Qatar, where she serves as the lead PI. She's made significant contributions to clinical genomics research and has interest in complex metabolic disease, population-based research, cohort design, and development of screening initiatives. She works closely with colleagues, not just in Sidra Medicine, but also the Qatar Precision Health Institute, Hamad Medical Corporation, and other partners within and outside of Qatar as well. She is instrumental in developing genomic sequencing programs for newborn screening in Qatar, focusing on detection of treatable disorders, both monogenic and polygenic. And she's interested in the genetic and development of genetic and polygenic risk scores for screening diseases of national importance to Qatar, particularly in the realm of type 1 diabetes. So today she'll be sharing with us her thoughts on going beyond single trait analysis, uncovering the complexity of type 2 diabetes risk through multi-GWAS discoveries and polygenic risk scores in a unique population. Dr. Akil.
Thank you very much for the organizers for giving me the opportunity to speak today. My talk has different flavor, Middle Eastern one, and you will see several slides that it could explain what we're doing, not only in terms of the polygenic risk score for type 2 diabetes, which is the focus of this presentation, but also about what we could offer for the International Scientific Committee in terms of cohorts that we have, since this event is dealing with cohorts and how we could take it to clinics. So I don't have any conflict of interest to disclose for this talk. And my talk will be around Sidra Medicine as a pediatric hospital, what we are doing, Qatar population structure, and how this will be a center for our research and also clinical translation, and also development of the risk score for type 2 diabetes and associated traits. So this is very brief about me. I studied, I don't know, everywhere. Okay, so, and I'm now working at Sidra Medicine. This is my team. I usually thank my team before I start any talk, because people used to put the team at the last slides, and usually I put them at the beginning before anything else. And without them, I won't be able to be standing here speaking about what we are doing to everyone. So I want to thank them as well as our leadership as well at Sidra Medicine and in Qatar in general. And this is Sidra Medicine, the place where I work, a state-of-the-art healthcare, children and women hospital. And it is one of the well-recognized healthcare providers in the Middle East. And what we are doing as Sidra Medicines, we provide patient care, we do medical education, and we also provide precision health research, and this research, it is under the umbrella of Precision Medicine Program for the country, which is probably you will hear more details later in this, and probably later during the conference from my colleagues coming from Qatar as well. So what we are, the 4 pillars are the programs at Sidra Medicine. What we do screening strategies for patients, and we also do report studies. And then advanced diagnostics and personalized therapies, it is what we are looking for at the end of having all of these infrastructure and patient cohorts recruited for our studies. So at Sidra Medicine, we do research for every patient. So patients who walk into the hospital, we do have around 15,000 visits per month. We recruit them to our cohorts, and we also recruit those who are not sick, who comes as a family members to our programs, and that's mainly focused on the polygenic diseases, rare diseases, maternal health, and also mental diseases. We have a neurology program dealing with several diseases that is also important nationally to Qatar and also to the Middle East region. This is very quickly about the focus of what I'm doing with my team. I just mentioned earlier, we do several disease focus area, type 1 diabetes, type 2 diabetes, and obesity. And of course, we do have a cohorts well characterized for each one of these disease areas, plus a very well established, just started newborn genome sequencing for screening for polygenic, monogenic diseases, which is I will give some information about a little bit later. I put this slide so people will see the cohorts that we have and what data we have for them right now. These cohorts, like I'm dealing with, I'm managing and I'm leading the two, one of them in the middle and the other one with my colleagues back at CIDRA. So we do have whole genome sequencing and electronic health records for Mendelian disorder research clinical cohort, autism genetic clinical cohorts, type 1 diabetes cohort, and also type 2 diabetes, which is a sub-cohort from Qatar Biobank that my talk will be focusing on for some polygenic score findings. Of course, this slide that I just mentioned, it is to open your eyes about what we have. Probably you will be looking for collaborations, and we are very open for that in terms of the available patient cohort in a very well characterized manners and unique population, just like the Singaporean population. We work closely with Qatar Genome and Qatar Biobank, which is now under one entity, which is called Qatar Precision Health Institute. And without this collaboration, we won't be able to see some nice findings, not only with me, but with other people who works over there. This slide, I like this slide a lot because it is showing the Middle Eastern society or the region that Qatar is a small country, but powerful in terms of the representation of this part of the world. Underrepresented, of course, Qataris and also Arab expats community who lives there and work there and represent a very unique collection of people who could be really contribute globally to the advancement of the precision medicine in general. Of course, there is another body who is funding our research, which is Qatar QRDI, Qatar Research Development Institute, and Qatar QNRF, Qatar National Research Funds, who are responsible on this. And of course, for my research, I have other international funders who are JDRF or Breakthrough TUND currently. The objectives of this talk, now I will be speaking a little bit specifically about type 2 diabetes, polygenic risk score, and associated traits. The objective is to explore the development and application of the polygenic risk score derived from multiple GWASs in predicting type 2 diabetes in this unique population. Of course, we have the focus on the enhancing the clinical outcomes through the accurate risk stratification. The objective is to evaluate the effectiveness of the multi-trait polygenic risk score in capturing the full complexity of the disease and to demonstrate how this could be adding value for the prediction in this unique population. This is just a quick, I know there is a lot of students and trainees here, so I thought maybe we put a slide about what its risk score is. Which is we computed from the GWAS studies using Qatar Biobank data. So the data that you will see here, it is based on Qatar population biobank. So it is a biobank cohort, which is we identified type 2 diabetes cohort, a sub-cohort from out of it. So how we calculated this, of course, there is steps to calculate the risk score, starting by the development, the base cohort, which is required, the base cohort, and then testing the performance and also evaluating it again in different population, we did different population, also different cohorts that we are used to evaluate and validate the outcomes. Starts with data acquisition, quality controls, developments, validation, and also recalculation of the PRS to see the performance rate. The case control studies that we are doing, it is based on the finding that we have, it's based on case control. And this is just a simple slide to explain the difference between case and control and how we could just use it to stratify the risk and move forward. We used the Qatar Biobank cohort, which is more than 15,000 individuals, and the whole genome sequencing data, which is we used to generate the GWAS of 80% of this cohort for the PRS development, for the refining, and then 20% for the development. And then this, of course, as a type 2 diabetes cohort, is a disease based on the HbA1c level. That's the stratification of the phenotypic data that we used. So individuals who are more than 6.5 HbA1c, they are classified as type 2, and those who are less as a control. And of course, we have thousands of those who are diabetics, apparently, in the population and that's why the disease is very national important, and solutions required in terms of the quick or fast or accurate stratifications in terms of using the genetics background of these very specific individuals. We used what's available with us in terms of the traits around the type 2 diabetes as a disease. And we all know that the BMI, waist to hip ratios, C-peptide, insulin, all of these traits, they are works around the disease and the incidence and the prevalence, and also the severity of the disease is related to a collection of these, but how we didn't know much in terms of the genetics. So we use these clinical traits to work around the risk scores of type 2 diabetes. So at the beginning, we developed the risk score for type 2 diabetes itself, and then another traits. Each one of these traits has its own risk score. And we found that the amount of these data show here that the main one, it's the T2D itself as a main contributor, age, BMI, well, all of the other C-peptide, which was surprising, contributed nicely to the disease severity. I want to show this slide here, and probably I have it in another location just to show the structure of the Qatari population. We do have, just like explained in the Singaporean population, we do have sub-ancestries in the Qatari population. And this data has been generated for the risk score study, which is I'm presenting, although it is published as well from one of our colleagues, using the genomic data of the biobank. We do have the Peninsular Arabs, the main Arabs in the region, who are in Qatar as well, 67%, and the other population, South Asian, African, and others. And we do have the type 2 diabetes cases, which is identified through the biobank, represented very well across all of this subpopulation and included in the population of Qatar. And that's something like assuring, because this is like, it's a well-represented population. It's not only the one that it is with one subpopulation, but not with the others. Of course, we used an efficient mixed model methods for biobank scale GWAS. I will be a little bit fast in these things because in terms of time. all of these traits associated with the disease. We use the linear mixed model with the covariant, which is mainly age, sex, BMI, first, then PCs. And we analyze and we develop the polygenic risk score accordingly. There is traits that it has a moderate score, and the others have like high score, and sometimes we do have like the lifestyle explained in this families that we have. As you could see, there is multiple IDs in each family, and some of them, they are high score affected and some low score unaffected. And we think that the lifestyle with the healthy lifestyle, of course, could be managed or could be explaining the risk of type 2 diabetes among those populations. Same for the BMI GWAS. I choose only BMI GWAS and the HbA1c GWAS here. Otherwise, it will be too long for other traits. And here, the Manhattan plots showing like several important obesity-related genes, like the MC4R and the FTO, high prevalence in this cohort, just like expected. But we do have another new genes, which is like LX1B and TME18, who are also have a role in childhood obesity. That's what we identified. Of course, results is not yet published, but this is very preliminary data for what we have, although it is on the mid-archive yet. This is HbA1c GWAS. And similar, we do have the HK1 gene that it is related to hexokinase 1 gene, also related to glycolysis and diabetes development. All of these data confirming the accuracy of the traits that we use and the effects of these within the population. Same here, summarizing the effects of the GWAS variants. Each of these traits on the right side, a number of the SNPs, the best PRS calculated for each trait also listed, showing this, the steps of the development, which is still required further validation, and the difference between case and control, and the effects and the way that this risk score was able to differentiate between cases and controls in this population. Same here for HbA1c. And now we are integrating all of these risk scores, and this is the method that we used. Of course, there is 16 machine learning classifiers all together, put together to come up with the model and train the performance evaluation for this. This is the polygenic risk scores for each traits, and each one of them have a different model for developments. And then we ended up with a multi-trait polygenic risk score that we could use. Here's the data, the classifiers that we are being able to put together for a data sets of 14,000, nearly 15,000, with the features that I mentioned, training data sets and test data sets, which is 20 to 80%. Very last few slides. The extra trees classifier, it was the best performing. And as you could see, for the area under curve, and also for the sharp values. Similar for, we applied the same for the subpopulation of the original population of Qatar, and we still see the Peninsular Arabs at par. It is the highest performing comparing to others, although their area under curve is still good for the rest, because the percentage between control and cases considers for each trait, for each subpopulation. And of course, we are using for validation until the stage, that slide before of this, it was Qatar Biobank data, which is used 20 to 80% to validate and to develop first. And now we are using the UK Biobank data and Genes and Health cohorts for the validation. Of course, there is different way of validating this, and we are awaiting also for another set of UK Biobank data set that it is not overlapped with the one that we use for the development to generate the further development, further validation set beside the UK Biobank and Genes and Health. This is the steps that we are used to generate and what we are doing to validate in the UK Biobank. A last slide for me, for summary for this presentations, the polygenic risk score development summary we just put through this. PRS summarize GWAS's results and predict outcomes in clinical settings and also in disease screenings. 12 GWAS is used to identify the genetic influence of T2D risk in the Qatari population. A single trait polygenic risk score shows significant association with T2D but may not capture the risk 100%. Multi-trait PRS model enhanced T2D risk stratification, and that's going for further development. This acknowledgement to those who are contributing to the work that we are doing using the population biobank. Some of them sitting here, so thank you in person. And also for others who are contributing to the funding and also for infrastructure like Sidra Medicine and the QRDI. This is Sidra again, a place where healing starts. So if you have an opportunity to visit Qatar, please come to Sidra and we're happy to provide you with the tour to see what we are doing and what we have over there. And also, I will not leave the stage without inviting you all to attend our 10th anniversary of Precision Medicine and the Future of Genomics PMFG Summit 2024 in Qatar, organized by Sidra Medicine, and also in collaboration with all other stakeholders around the country. A great opportunity for learning and also for being part of these initiatives from the Middle East. Thank you.
Thank you very much, Dr. Akil, for a wonderful talk. So I think what we'll have now is a panel discussion. So I'd like to invite the speakers to take their seats over here and we can get started. Thank you. So there have been some questions that have been put up in Pigeonhole. So thank you everyone for your participation. So I'll just start with the first question. So let me just activate it. Right, so I think the first question is directed at Prof Wong. So the question goes, how would you adjust the architecture of your autoencoder for sequence and SNP data? And how can we use foundation models for analysis of smaller clinical or specialized data sets?
For SNP and sequencing, where you're determining the genome,
yeah.
So this one you will not have much batch effects to talk about, right? Because this is not quantitating. So the batch effect only usually affects quantitation. So if you're not quantitating, you don't have to worry about that.
Yep.
Yeah. So there's no need to.
So
it's not the correct tool for you to use.
Yeah. So maybe following a bit on this question. Do you see this, being applied to other areas? I mean, this is for single-cell RNA-seq. Do you see this, for example, like bulk RNA-seq? Do you see any need for this? Or?
Bulk RNA also suffer quite a lot from batch effects, but you probably do not need to use map batch star technology because of one. This is based on deep learning. Then sample size is very important.
Right.
And in single cell, I have thousands of cells to do the training. So you can do this very high precision correction or handling. But bulk experiment, you may not have the thousands of patients, right?
And so maybe going into this,
so you need some other alternatives.
Yeah. And so what I'm hearing is that because it's a machine learning or deep learning approach, then it's very data hungry, and so you need.
You do need more more training cases, although you can reduce it a little bit, there are things that you can do in the architecture of, let's say, my autoencoder. I just show you encoder, decoder, and explain the basics. But there are ways where you can let's say you don't have a few thousand cells, but less. There are actually things that you can do within how you architect the autoencoder to have less need for the thousands of cells.
Sure. And you've listed some applications like I think cancer, single-cell RNA-seq is one of them, and hematological cancers, right? And then the other one, no, sorry, that was a colorectal cancer.
So that is, they're all single cell. So
Yep.
in terms of what you do is more or less the same. But in this case, the two examples I've shown, the first was on multiple myeloma,
Yep.
so it's liquid tumor.
Yep.
And the second comes from colon cancer, so it's solid tumor. Third one I didn't show, actually is COVID patients.
Right. So that's is it immunological, immunology based, looking at?
No, we are just, someone happened to profile these patients
Sure.
using single cell technology.
You looking at the.
So we look at, for the COVID, so that one we are looking at which kind of patients would develop a more severe COVID case.
Right.
Because not some patients like me, maybe you get headache for 2 days and then we are fine. Some may have much more complications. So we are looking at whether there's any difference between these two kinds. Yeah.
Wonderful, thank you. So the next question is for Dr. Jimmy Lee. I think there's a lot of interest in this. So I think schizophrenia patients have high chances of multi-morbidity, such as metabolic diseases. So I think how can we use precision medicine to perhaps better predict, the schizophrenia patients that might have multiple comorbidities? Is this something that you think is a problem? Is this something that you're looking at?
So you're talking about cardiometabolic morbidities in association with schizophrenia patients, like in terms of prediction. So we do, actually clinically right now, what we do is we screen. So everybody with schizophrenia is presumed to be high risk, and we do yearly health screening. Some as early as 20s, we start screening and we start detecting, and we put them through treatment, the usual kind of guidelines in terms of treatment for high cholesterol and glucose tolerance issues. Yes.
Okay. And I think just to follow on that, I think another question is, do you have any thoughts on pharmacogenomics for other psychiatric disorders beyond schizophrenia? Are there any areas that you see would benefit from this as well?
In psychiatry, I think certainly. It's an interesting question to ask as well. So I have a colleague in the audience here who does PGX for depression. But I would say, one interesting paper actually came out some years ago, and what they found is that in patients with depression, the polygenic risk score for schizophrenia actually predicted the development of treatment resistance. Much better than actually the other kind of polygenic risk scores. Now, the question here is why is that so? Whether it's because the general genetic or genomic burden or disruption connotates something that's more biological, and hence, then the biological vulnerability connotates development of resistance, which is what I presented here and what we see in schizophrenia. There's also interest in terms of bipolar. Now, when it comes to pharmacogenetics, I think most of the current platforms focus on more pharmacokinetics, here the metabolism, right, the cytochrome genes and all, and the transport proteins. That is useful, but so far in clinical practice, it's not something that the clinicians think of using, because if you use it in terms of PK, then you should come with guidelines in terms of with this particular genotype, what does it mean in terms of dose adjustment, dose titration. If he's talking about, we don't have a very severe kind of fatal side effects that require screening, like carbamazepine and epidermal necrolysis kind of problems. So what actually clinicians will probably value a little bit more are PD genes, so pharmacodynamic kind of genes where I can predict response and resistance, because we suffer from multiple options when it comes to psychotropic medications.
Wonderful, thank you. So I think I'd like to move on and look at questions directed at Dr. Akil. So I think one of the questions is, so I'll just bring it up. So for the Qatar PRS projects, what kind of data platform was used to calculate the polygenic risk scores? Is this a, I think maybe to elaborate on the question, is this a commercial data platform, or is this something that was developed sort of like by your bioinformatics team? And how did you go about developing sort of like the, clearly it's a very intensive bioinformatics infrastructure. So how do you go about developing that?
Very good question. Thank you. Of course, as I mentioned, the data set that we have, it is the Qatar Biobank, which is used for this study. And the bioinformatics approaches that we use, it is all in-house. So we do have a very dedicated bioinformatics teams who are trained on utilizing the large data sets and on the generating the polygenic risk score in particular, who have been utilized to develop machine learning, which is available commercially, but applying them on these data and generate the findings that you just seen. Of course, this is not everything. There is a lot of other data that I didn't show in the background. So it is not commercially 100%. It is available machine learning tools, which is you all probably knows about, but who applying them, it is not industry or a company, it is us ourselves, and that's part of the capacity building that we have for the genome program in Qatar. So nothing is going outside the country to be sequenced or to be analyzed. It is all in-house, and that's how probably make us recognized over several others who are just like here and there. But definitely it is all in-house.
So maybe as a follow-on question from me, so you have a lot of whole genome sequencing data that's been generated. So you showed today sort of like the polygenic risk scores. To what extent are you seeing contribution from rarer variants that you're seeing, in the whole genome sequencing data?
Yes. Can you just last part? I didn't
Sorry,
hear.
so you have whole genome sequencing, which means that you're able to see not just the sort of like SNPs, polymorphisms, but you also see rare variants.
Structural
so
variants.
What's the contribution of these rare variants? Do you see them sort of like, do you see any value perhaps integrating them into the polygenic risk score with not just looking at the common SNPs, but also the rare variants? Do you see any contribution?
Yeah, that's a great question. Of course, this is mainly common variants, but definitely we do have a rare variants or like, let's say, unique variants, identified for certain segments of the genomes related to the population over there. It's not in this presentation, but I can mention about it here. It is mainly for the HLA segments, which is expected probably, but not have been discovered. We do have a certain set of HLA SNPs and tag SNPs and certain haplotypes that have been identified specific for type 1 diabetes cohort. And that's population specific, not yet identified before. This is very, I cannot say more than this because this is very unique data which will be published very soon, inshallah. Thank you.
And so you're identifying people with sort of like this on the higher end of the polygenic risk. So number one, I guess, is how is this being communicated back to the research participants, and how do you see this being sort of like, how does this become part of their healthcare management? Like, for someone with a high risk of type 2 diabetes, for example, who may not have developed diabetes, are there, do you have any strategies to sort of like manage their risk, and how is that?
For sure. I mean, the ambition of Qatar Genome and Qatar Biobank, it is beyond sequencing the data and presented in a conference, of course. This is a big national entity initiatives, and there is a lot of plans, and that's all within Qatar Health Strategy 2030. So returning the results is actually before returning the results, incorporating the results into electronic medical records. It's one of the objectives of this study. But I haven't explained anything about this because it's another objective, a standalone project, which is second in the line to the development of the polygenic risk score for the associated traits. So incorporating into electronic health records, this is what we are doing currently, but that will be also going through the national strategy, how we could return the results to the biobank participants. The Qatar Biobank directors are sitting here. They could probably speak about
and
this
so maybe
better
just
than me. Yeah.
for my education, I mean, in a lot of places where this is being done, especially when we're talking about integration into electronic health records, there's always, does it affect the insurability of the participants and all that? So maybe I'm a bit ignorant, but is this not a concern in Qatar because the healthcare is
strong enough?
Yeah, so
and
there's no
well-maintained,
there's no like private insurance or not so much like people have access to healthcare, and so there's less worry about insurability being affected. Or is there a legal protections?
There is a legal protection and there is insurance as well, but this is all covered by the government, and this can be returned to the results. And in the biobank, we do have like a second time point, like a follow-up. A second time point collections for the participants who are collected at the first time recruited, and we do have a couple of thousands of them already recruited and already in communication with the biobank about if any findings. And we also are putting in mind that we approach participants, and some of them have been approached for another cancer, breast cancer studies managed by other hospitals in Qatar, returning the results and put them on the pharmacogenomic studies and also treatments, and many success stories based on that, definitely.
Okay, wonderful. Well, I think that's the about the amount of time that we have. So everyone, please join me in thanking our panel of speakers for their talks. Thank you very much.